Good afternoon, everybody. I would like to start by thanking the organizers for inviting me to this session. And I was invited because of the publication we had last year on inferring gene disease associations that were published in PLOS ONE, and for the metadata work that we did surrounding it. And I would really like to share with you today the experiences we had in creating and working with this data set. Just by introducing the problem that we all know, that's the biological complexity and data overload that we face. And this graph is actually now about 10 years old from a paper in 2008 on clustering of diseases. And basically what we thought about as one solution to dealing with this was like this problem of information overload was to create a literature-wide association study. So it's basically a text mining pipeline. So it starts with a controlled vocabulary of integrated dictionaries. It goes through a Medline abstracts of about 17 million from 1980. We index all the concepts per abstract. We compute the co-occurrences, frequencies of these concept pairs. And we then build concept profiles from this. So for any concept, that would be a list of all other concepts that are related to it. And it's weighted by the uncertainty coefficient of the co-occurrence frequencies. So we have a minimum of 5 abstracts for creating a concept profile and a maximum of 140,000 concepts represented in the profile. So this would allow us to actually expose associations independently of the documents that they come from. And when we do this, this will allow us to actually do implicit information matching. So concept profile matching is what we call it. And what you see here is like 3 different representations of how we do this. So here is represented as a small graph where we have intermediate nodes, and then we infer the link between these Z and the X nodes. So as I said, we represent them as a vector of related concepts. So we weigh them by the uncertainty coefficient, and then we can match them using different vector matching methods. And there are a lot out there, but we use the mutual information score. So then we can actually infer these implicit links, and looking at these 2 concept clouds of a gene and a disease, we can rank and say how important this gene-disease association is according to the matching algorithm. Now, how much knowledge do we actually capture with this method? Well, if you see here this small red square here, this is the explicit knowledge that we capture. So these are all the co-occurrence pairs of the gene disease associations that we find in the literature. And it's about 1.5 million. And we capture around 204 million concept pairs, exposing these associations to our method. But you actually see that the potential knowledge space is more than double. So this means that there is definitely, of course, room for improvement in adding other type of data sources, and to really, for rare diseases, for example, where you don't have a lot of information already published, like how do you get to this type of information? Anyway, this was published in PLOS ONE last year, and I'm not going to focus on how we benchmark and the results of this paper, because this is a metadata and indexing session. But if you're interested, please go and have a look in the paper. What I just wanted to show you was that one example of such an association and how we can think about using this technology to rationalize for gene disease knowledge. So the CYP2R1 had a strong association with the sloth syndrome. And the connecting concepts that you can see here was that this gene, DHCR7, was one of the connecting wire concepts. And they actually defects in this gene actually causes sloth. And we also had 7-dihydrocholesterol as one of the connecting concepts. So the reasoning that we can apply to this information is that, well, we actually had the pathological hallmark loss, and 7-hydrocholesterol is a precursor of vitamin D3, and defects in this CYP enzyme is known to affect vitamin D3 levels. Thus, this LVAS association implicates CYP2R1 in loss, since defects may potentially lead to 7-dihydrocholesterol accumulation. And yeah, this is just an example of how you can use this information to rationalize a gene disease association. Of course, I wrote these now as just words, but they are all ontological terms for clarity. And now I would like to switch gears, and I would like to tell you about enduring the pain of FAIR. We heard a lot about FAIR this session, and especially about enduring the pain of FAIR without metrics. So the actual research time that this took was about 3 years with our team to, developing the pipeline and doing all the analysis and benchmarking and interpretation and so forth. The verification took 1 year. And I say this because I think it's great, that we do all these efforts. We have to do it, but we can't forget how much effort there is at this moment to do this type of modeling of the data sets. One thing contributing to this was that, we published in February 2016, and in the FAIR Guiding Principles paper came out in March 2016. So we were just about, we were following the Force 11 guidelines. So we had, of course, idea on how to do it, but it was not really so much on the way. And then, of course, a year further, we had a Lawrence workshop, which is like a collaborative workshop on how to make data fair for open science. And coming out of that was the FAIR Metrics group that was already mentioned by Susanne, and they are working on how to come to metrics so that we can better know how to deal with these type of data sets. I'm not going to go through all of them. I just want to say that if you take the FAIR principles and you look at it from a metrics point of view, the red ones are those that you can say a yes or no to, and the yellow ones are extensible, so you can apply a number to it. The blue ones are a few ones here that they are not really know yet what to do with. And let me see, just to see what we did at that time without knowing all these metrics and just have an idea of the principles, was that we created this sort of code and data overview. So this diagram was created by Mark Thompson in our team, and it represents, like every link in this diagram is a link to a data set, a result, an algorithm, where it is available. So you can just explore the paper in a way in this sense. And the repositories and licenses that we used at that time was the data dryad, because it was published in +1, and +1 is the recommended data repository. But we also repositoried in the B-Hub, which is the Dutch, one of the Dutch national repositories for data, and that's more for our own use, so that we can actually browse and create a catalog structure for this data. All the code decisions we took was to put everything in GitHub and to put a general public license on all the data. All the figures are also in there, and they have a general public license. The Melody and Thesaurus, they are their own license, or they have the National Library of Medicine license. And these are things that, you can say we took these decisions, but it took quite some time to come to the right, what sort of license should we actually use for the type of data that we are working with. And those are things that take time. And of course also the type of where you want to, what's the type of data, where do you want to reposit it in. So that is, well, at least our decisions that we had, that we took at that time. For data exploration, we also modeled all these genesis associations as assertions according to the nanopublication scheme. So all the number publications are available, of course, at the Data Dryad and so, but also at the rdf.biosemantics.org. And I don't know how many of you can read RDF in the room, but this is at least to give you an idea that everything, all these nano publications, they are all also linked with a unique identifier and all the concepts in there that you can explore. Another thing that we did, because this is of course for humans not really very usable, but except for data scientists. But we also collaborated with the Scripps Institute on the knowledge.bio application. So they created an application where they loaded all of semantic Medline, and we loaded also part of the nano publication dataset into knowledge.bio. So you can get knowledge.bio on Bitbucket. This is an MIT license. It has a database and a web interface, and you can, that's more of a human exploration, what you can do. Although, if I would have been a year further, I might have been more interested to look at the fair data point. So, where you can actually put your CSV data, your metadata, you can create your models, really the deeply modeling of it that we did with our data, ontologies, identifiers through the verifier tool, create the RDF file, and actually expose it to a fair data point, which is, I would say, complementary to CDAR. I think CDAR is really on the deep metadata, so a lot, and a fair data point is really on the minimal data metadata. So yeah, they're definitely complementary in that sense. Another thing that we did was to ensure sustainability. So of course we put every code and everything in a public repository, but we found that it's also really useful actually to work with private companies, because there with them we did with the Euretos, we did a knowledge transfer. So we work in collaboration with them to actually implement the algorithms that we developed into their data platform on knowledge discovery on gene disease associations. So here you see the gene and the disease and how they implemented our algorithm in their interface for their clients. So we have a MOU with them, a memorandum of understanding, and they also actually act as sponsors for our research. So yeah, it's a two-way communication and it's another way to make sure that your results stay, but then in the private domain. So I guess what I would like to go for you to take with you from this session is that the concept profile technology, it really covers about half the potential knowledge base for gene disease associations. So of course our group is working now to figuring out like how can we access the second half and algorithms and data to work with that. And doing reasoning over fair data points is really something that we have in mind for the future. And I would also like to say, and with that, verification, and especially in a collaborative setting, is really needs the expert knowledge. So I come really more from the bioinformatics, text mining background, and I worked very closely with the biologists and the fair data experts like Mark Thompson and Rajaram and Casey in our group to how to expose this data in the best way. It also requires soft skills because you need to be able to communicate and how to represent all this data. It's really time consuming, especially the first time. So for us, it took about 25% of the research time. Although I'd estimate if we do it again, it would take around 5% of the time actually, because if you did it once, then a lot of decision path can be cut short. But it also really leads to further collaboration. If you do it right, if you expose your data in a good way, you can take advantage of people like, that are interested to work with your data, like the SULab people that wanted to integrate it into that knowledge base, and we're still working with them now. Also, we are still working with UPM, so the Wilkinson Laboratory, on citation research based on our data sets and on Euretos for the knowledge discovery applications. And last but not the least, it really gives a very satisfactory feeling of doing good science if you take the time to really expose and share your data in this sense. So I'd like to end with acknowledgements of all the people involved from the LMC, Leiden University Medical Center in the Erasmus, where a lot of the algorithm knowledge started actually in the text mining pipeline, from the Scripps Institute and the many funding organizations that funded this work. So, thank you. Of course, Marco is it's a group leader, so he's, I should have pitched your talk, I'm sorry.